plant growth-promoting microorganisms in coffee production, from isolation to field application. In this presentation, our goal is to offer you a quick summary of the research and conclusions. Please follow the links to review the research in further detail. Thanks to all the authors and publishers for making this research public, thereby advancing science and innovation. Coffee is an important high-value crop because its roasted beans are used to produce popular beverages that are consumed worldwide. Coffee plantations exist in over 70 countries and constitute the main economic activity of approximately 125 million people. Currently, there is global concern regarding the excessive use of agrochemicals and pesticides in agriculture, including coffee crops. This situation has motivated researchers, administrators, and farmers to seek ecologically friendly alternatives to decrease the use of synthetic fertilizers and pesticides. In the last decades, multiple studies of the rhizosphere at the chemical, physical, and biological levels have improved our understanding of the importance of beneficial microorganisms to plant health and growth. This review aims to summarize the state of the use of plant growth-promoting microorganisms, PGPM, in coffee production, where the most extensively studied microorganisms are beneficial plant growth-promoting rhizobacteria, PGPR, and arbuscular mycorrhizal fungi, AMF. This review also contains information on PGPM in regard to plantations at different latitudes, isolation techniques, mass multiplication, formulation methods, and the application of PGPM in nurseries, monoculture, and coffee agroforestry systems. Finally, this review focuses on relevant research performed during the last decade that can help us improve sustainable coffee production. Plant growth-promoting microorganisms that are associated with coffee are important components of microbial biomass under monoculture and agroforestry systems in all geographical areas where coffee is cropped. In particular, AME have been associated with clear improvements in nutrient uptake and tolerance to water stress by coffee plants, which are ecological and economic importance. Moreover, plant growth-promoting rhizobacteria can support increased nitrogen fixation, which is especially relevant for the growth of coffee plants. Nevertheless, there are only a few studies analyzing the indigenous communities of these microorganisms in coffee plantations, even though coffee is an important commodity in over 50 countries that are involved in its production, trade, and consumption. Based on the above, PGPR and AMF are potential resources for bioinoculant production, especially considering the use of indigenous soil microorganisms that are adapted to site-limiting conditions, thus increasing the functional characteristics of microorganisms that improve sustainable coffee production.